Hi everyone, I'm April and I'm thrilled to introduce the rest of our research team, Adarsh, Ivy, and Merab, where we will be diving into our project titled Discovering and Optimizing Drugs to Inhibit EZH2, Applications in Treating Cancers, with our efforts being guided by our mentor, David Candace. Ivy will now begin.
We will begin with an overview of the enzyme EZH2. EZH2 is a subunit of the PRC2 complex which is involved in gene silencing. It catalyzes the trimethylation of H3K27, which are DNA modifications that function in transcriptional repression. However, as you can see in the diagram above, the overexpression of EZH2 leading to increases in H3K27 can cause repression of tumor suppressor genes, thus proliferating tumor cells. Furthermore, EZH2 is involved in the regulation of cell cycle progression. The dysregulation of EZH2 accelerates cell proliferation and supports cancer stem cell survival. EZH2 inhibition allows cell apoptosis and arrests cells, allowing for elevated levels of tumor suppressors. Recent studies, as shown in the provided clinical trial graph below, have found repetitive patterns of EZH2 activity in various cancers, including prostate, breast, bladder, etc., making EZH2 inhibition an attractive target for anti-cancer drugs.
EZH2 inhibitors are important because they suppress tumor growth by stopping the methylation of the H3 histone. They can also stop protein-protein interactions of PCR2 that are EZH2 dependent. EZH2 inhibition can also improve the efficacy of other cancer treatment pathways. EZH2 relies on the S-adenosyl methanoate molecule to function. This SAM molecule is a cofactor and is a methyl group donor. After donation, it leaves as S-adenosyl L-homotosine SAH, a natural inhibitor of methyltransferase activities. There are two main categories of EZH2 inhibitors. 1. SAH inhibitors that increase SAH levels, and 2. SAM competitive inhibitors, which occupy the SAM binding sites, preventing methylation. An example of this is tazemetostat, which is the only FDA-approved EZH2 inhibitor.
Given previous research on EZH2 inhibition, here are a few challenges faced during trials. First, short half-life in vivo, which means that the drugs are rapidly metabolized and require higher doses to achieve desired results. Second, low efficacy. Genetic, racial, dietary, gender, and other extraneous variables may cause the drug to be ineffective. Third, drug resistance due to genetic mutations or adaptations in cancer cells will also decrease efficacy. Lastly, residual H3K27 can persist due to the activity of EZH1, as it has been evidenced that EZH1 methylates H3K27 in the absence of EZH2. However, one method to combat the methylation of H3K27, as shown in the diagram, is the dual inhibition of EZH1 and EZH2 to address the residual trimethylation and overcoming resistance with single-agent EZH2 inhibition.
Traditional drug discovery is a lengthy and very costly process, often taking 10 to 20 years for clinical trials, FDA approval, and actually getting to the market, costing between $500 million to $2.6 billion annually in the US. And it frequently struggles with the complex and changing nature of proteins we have in diseases. Computer-aided drug design, otherwise known as CAD, offers this modern-day solution using advanced computer simulations to predict how drugs will interact with these proteins, which speeds up the process and reduces costs by allowing for more efficient virtual screening and precise modeling. The two general types of CAD approaches include structure-based and ligand-based drug design. In structure-based design, we use the 3D structure of the target protein to find molecules that can bind to it, using techniques such as molecular docking or de novo drug design. In ligand-based design, we use the known active compounds to identify new drugs through methods such as QSAR modeling and PK/PD modeling. By employing CAD in our project, we leverage its vast potential to explore different strategies, maximize efficiency, and increase the likelihood of finding effective EZH2 inhibitors. And here we post a research question to put the test on. Can machine learning models be used to identify EZH2 inhibiting drugs, ultimately creating novel therapeutic agents for cancers? Here's a concise overview of our planned methodology as shown on the slide. We started with a comprehensive literature review and data collection using key datasets such as CHEMBL for chemical profiles of inhibitors and the protein data bank for 3D molecular structures of EZH2 to gather essential information of potential drug compounds and their targets. After processing the raw data, we used PADEL to compute molecular descriptors and employed regression models to predict biological activity, otherwise peak 50 values, quantifying the chemical characteristics and the potency of these compounds inhibiting biological functions. Next, we use Swiss-stock for molecular docking to stimulate interactions between inhibitors and target proteins, predicting how well our drug can bind to EZH2 proteins. Finally, we conduct statistical validation and visual analysis to ensure our predictions are accurate and meaningful. This entire process was implemented on Google Colab for efficiency and collaboration.
Okay, the first step in making our model was data collection and pre-processing. We pulled potential EZH2 inhibitors, really any compounds that interacted with EZH2, from the chemical database, and that left us with around 1,500 data points. Then we classified them as active, inactive, or intermediate based on their IC50 values, which is essentially a measure of inhibitory potency. We also implemented a data cleaning function to remove any points with faulty or non-existent values. Next was exploratory analysis. We visualized current existing data, removed the intermediate class for plot simplification, and calculated Lipinski descriptors for each of our compounds, which are 4 different characteristics of a compound that determine its drug likeness. We updated our data frame to include these descriptors, and you can see that they are molecular weight, log P, which is lipophilicity, number of hydrogen donors, and the number of hydrogen acceptors. We then plotted our Lipinski descriptors against the bioactivity class of each compound and performed Mann-Whitney U-test to determine statistical significance. Ultimately, each descriptor except the hydrogen donors and hydrogen acceptors proved to be significant in the case of our set of EZH2 inhibitors. After exploratory analysis, we utilized Paddle software to calculate the molecular fingerprints of each compound, which is like a binary code to represent a compound. We then removed low variance features and put the fingerprint into an x-axis data frame and the PIC50 values, another form for potency measuring, into the y-axis. We split the data into 80% for training and 20% for testing, and then used the random forest regressor model to predict the PIC50 values for our dataset. Results can be seen on the right. After using just the random forest regressor model, we used lazypredict to rapidly generate 42 models and compared each of them using statistics like R-squared and RMSE, which both help us evaluate model accuracy. We identified the Gaussian process regressor as the best model and then isolated and retrained it on its own. We ran the model on testing data and compared its predicted PIC50s to the actual PIC50s, and then we calculated various statistics to evaluate the model. Its R-squared ended up being around 0.75, its RMSE 0.8, the mean absolute error 1.75, the average percent error about 33%, and the median absolute error about 1.5. Ultimately, the model was not super successful because when it comes to predicting things like PIC50, a value that functions on a logarithmic scale, you need to be extremely precise with your predictions for your model to be considered actually useful. But now, let's talk about some limitations from our project. First, we had a limited dataset. Second, we only predicted one aspect of each of our compounds, which was drug potency. Third, we removed low variance features due to a lack of computing power, and this likely decreased accuracy. Fourth, the lazy predict library isn't perfect. More rigorous testing of each specific model may have yielded different results. And fifth, we did not predict the drug's interactions with anything other than EZH2, so each compound may have had other side effects in the body that we do not know about.
Using Swiss-stock software for molecular docking, we analyzed the binding affinities and their poses, as we were able to now validate how well our compounds fit into the binding sites of different EZH2 proteins and the strength of their interactions, suggesting that our compounds could effectively inhibit the target proteins and their biological pathways. Despite some of the limitations with in silico and the need for further in vivo and in vitro testing to completely validate our drug compounds, deep learning models like graph neural networks, multimodal models, and variational autoencoders could show significant promise in drug discovery. Unlike traditional machine learning, deep learning can automatically learn complex patterns from large datasets, revealing intricate relationships between molecular structures and biological activity, where we hope to actually implement this in our project in the future. In conclusion, our project has deepened our understanding of advanced computational techniques used in drug discovery, and we look forward to continuing this research to contribute to more personalized, cost-effective, and efficient cancer treatments. Thank you everyone for viewing our presentation. Let us know if you have any questions or comments. Thank you.